All right, so my name is Colin Lamke, and I'll be introducing Dr. Michael Gore today. And so Dr. Gore got his bachelor's and his master's from Virginia Tech, and he received his Ph.D. from Cornell. And he's currently a research geneticist for the USDA-ARS, and is an adjunct faculty member with the University of Arizona at Maricopa, Arizona. And so his research interests include complex trait dissection of nutritional and stress-tolerant traits. And today, he's going to be presenting on exploring the genetic architecture of traits in maize and cotton with a next-generation platform. So please join me in welcoming Dr. Gore.
Thank you so much, Colin. I just want to say that thank you for the opportunity to give a talk today, and that the University of Nebraska has a fantastic group of graduate students. And I would like to pause and give them an applause for organizing everything right now. So, unlike Jody, I felt inclined to put the word next generation in my title so that they would not retract my talk. So if you're wondering where the small town of Maricopa, Arizona is, which is what I was wondering before my job interview, it's about a half an hour south of Phoenix. So if you ever want to come there, it's a great time to visit. Just please don't come in July or August or you'll be cursing me. And this, I think, was one of the first times I've ever traveled from Phoenix to someplace else where I had gotten off the plane and it was actually hotter. So that's a first for me, and it's very strange, and it kind of scares me. But without further ado, where are we going with this today? So the first thing that I'm going to talk about today is whole genome patterns of DNA variation in maize. So it's kind of broken up to 3 vignettes: the genetic architecture of carotenoids, seed traits in the maize Namm population, and our attempt at field-based high-throughput phenotyping of cotton. So just in this most simplistic form, which let's just concentrate on the genotype. And so we say we have 6 inbred lines here, and say we have done resequencing of a candidate gene. So we have 6 different PCR amplicons that have an alignment here. And the most rudimentary variation that we can think of is single nucleotide polymorphism, SNP. And here we have an AG allele. So if you think of maize, so just any 2 maize inbred lines, the divergence between those maize lines can vary between 1 to 1.4%, which is pretty close to the same rate between human and chimpanzee. If Ed was up here, he would be showing a picture of a chimpanzee. So that is about 14 times higher than that of human beings. So maize has tremendous diversity, and it's on average like 3 to 5 times higher than most grasses out there. And I'm sorry, Steve, that also includes wheat. So now we want to think about the functional diversity in the maize germplasm. And here we have the phenotype. So this is just variation for kernel color, and this is highly correlated with total carotenoids. So on the far left here, we have white maize. Whoops. Should point the right way. So we have white maize, and this is yellow, and over here we have orange. So the transition from yellow to white, or actually I should say from white to yellow, is basically one gene called Y1. But the transition from yellow to orange is mostly explained by 5 common loci, and this is something that we've recently have tested in 10 families of the maize Namm population. And I will be talking at length on the maize Namm population as I go forward. So how do we connect genotype to phenotype in maize? So how have we been doing this for the past 5 years with the maize Namm populations? So first and foremost, going from the phenotype to the genotype, we want to think of linkage analysis. So this is just in like a simple family structure. Say we have P1 and P2, and we have crossed them. Just say we have 1 generation of recombination, and we're now QTL mapping into F2 population. One thing to point out here is because there hasn't been very much recombination in this family history, that we're going to have large recombination blocks. So that gives us low resolution. So less markers are needed to capture the recent recombination, but this is at the expense of lower resolution. And now this is probably a common term that we have all heard of. It's transferring an approach called genome-wide association studies from humans to maize, and now it's filtered down to every other crop. And this is, in a sense, you can think of this in mapping complex traits in a population that you created from, say, some of these valuable germplasm lines, which were talked about previously for wheat. So we can take advantage of a lot of the ancient recombination that has happened through the history of creating these populations. So this affords us high resolution mapping. So in a crop like maize, LD, so the non-random correlation of alleles from two loci, decays very rapidly across the genome. On average, it breaks down to about 2 kb across the maize genome. So this gives us great resolution for mapping complex traits. The trade-off, though, is even though we have high resolution, we need millions of markers for GWAS and diverse maize. So that's just one trade-off. And you can kind of view this now as going from the genotype to the phenotype. So how are we going to integrate family mapping in GWAS into a platform that's powerful and can be used as a community resource? And this is where the maize Namm population comes into play. And I'm sure most of you in this room have heard of the name of the maize Namm population. So it's taking 25 diverse lines, which capture about 80% of the common SNP variation in the maize germplasm pool. So some of the great maize breeders at North Carolina State University created a highly diverse global panel of 300 maize inbred lines, and through genotyping, they chose about 25 diverse lines that captured, some of that common variation. Now, one thing to point out, the maize Namm population is not capturing all of the genetic diversity in the maize germplasm pool. There are a lot of other breeding pools out there, breeding populations out there, and a lot of other rare alleles, if you will, that are not being captured. So that's just a very small caveat here. So they were all crossed to B73 and a common reference design, and we already had somewhat of a history from Jody about the creation of B73 out of Iowa State, and that was chosen because it has had such a tremendous impact on both private and public maize breeding populations. So it is found in a large proportion of the pedigrees of common and elite inbred lines, and also probably greater than half of the parents that were used to create the maize Namm population were tropical. So there's a big issue with the confounding of flowering time. So by crossing to a temperate line, that we were able to create these recombinant inbred line families that could then be grown elsewhere, such as like Cornell and North Carolina and all across the Midwest as well. And this is what we call the Namm population. So this is referred to as nested association mapping. And here we have the common parent B73, and there are 200 recombinant inbred lines per family. So for the 25 families, that totals 5,000 recombinant inbred lines. So all of you graduate students thinking out there, if you're complaining about having a population size of maybe 300 recombinant inbred lines, now you kind of know our pain for the past 5 years of having a population size of 5,000 recombinant inbred lines, but it's a great resource nonetheless. So how are we genotyping Namm? So one really cool aspect here with Namm is we use a common B73 parent for all the crosses, okay? So that means we can genotype these rare SNPs so that they will be polymorphic in all of the 25 families, okay? So that allows us to, in a sense, like QTL map using the recent recombination that was, in a sense, generated by creating the 25 Namm families. But the real power of Namm and being able to use the ancient recombination requires next generation sequencing of the parents, okay? So by doing next generation sequencing of the parents, we can then project the haplotype onto the 5,000 recombinant inbred line progeny. Because so you're probably asking how can we do this? So with these common SNPs, and there's about 1,200 common SNPs that were mapped in the 25 recombinant inbred line families, that allows us to track the chromosomal segments from each of the parents. Okay, and then after we do next generation sequencing, we can, in a sense, like overlay, or some of the terminology we use is to project that information from the founders, the 27 parents, onto the 5,000 recombinant inbred RILs. So that's 183-fold cost savings in genotyping. So just to construct a Namm map, thousands of markers were scored with a SNP chip. And for GWAS, we need millions of markers scored on parents by next generation sequencing. So that's, in a sense, to really utilize an ancient recombination that has happened in the history of the population. So how did we do this? That's what kind of this part of the talk is. So one thing to kind of give you a viewpoint of this, when I began my PhD in 2004, we began sequencing like single candidate genes. But towards the end of my PhD in close to 2009, we began able to do whole genome resequencing with some of these next generation platforms. So in that short amount of time, we were able to jump from just like a candidate gene approach to the whole genome. So that totally changed how we approached our science. So here we use an approach that basically just targeted the low copy fraction of the Maize genome, because the Maize genome is highly repetitive. So it's about maybe 80 to 85% highly repetitive. But at the time, we couldn't just do whole genome sequencing of like all these 27 parents. So what we did is use an enzymatic approach to just cleave preferentially into low copy fractions of the Maize genome, and then we took these libraries, if you will, and then that's the target that we use to call SNPs. So with this next generation sequencing platform, we were able to call 3.3 million SNPs and indels. As we all know, Maize is an ancient paleopolyploid. It was once a tetraploid. So 1.4 million mapped to a single locus. So by using a Fisher's exact test, we were able to kind of tease apart which SNPs were the result of parology and which SNPs were actually real. In a sense, they mapped to a single position in the Maize genome. And only the 1.4 million single locus SNPs were used to calculate what I'm about to show you now. And an error rate for the 1.4 million SNPs was very low, about 1 in 2,500 base pairs, and about half of those are resulting for parology issues. So for us to get down to like a set of SNPs that were, you know, very clean and all like true positives, we had to use machine learning approaches to, in a sense, precisely call these SNPs. But when we were actually doing that, what we found out is we were throwing out a lot of the rare SNPs, just so we could get a set of ones that were real. So this is a comparison from Selective to Sanger sequencing here. And so this would be like with a candidate gene approach. And as you see here, most of the SNPs are rare alleles. But with our next generation sequencing platform and pipeline, we're excuse me, we're throwing out a lot of these rare SNPs. So one important caveat here is that it's very important to improve calling for trying to actually test these rare SNPs for complex traits. One, I would say this is maybe one of the more unusual findings that we found with this HapMap project, that in additional low copy sequence not found in a reference B73 genome. So on average, other line has an additional 7.8% not found in B73, and this would be, in a sense, like our positive control. These are B73's reads that are not found in the draft or the result of sequencing errors. So what we can kind of draw for this, if the unshared fraction follows the neutral SNP distribution, B73 and other may only capture approximately 70% of the low copy sequence across these 27 lines. So what that means then is, okay, we have one single reference genome sequence for B73. That now argues for us needing more, because there's a large amount of like the low copy fraction, which includes genes that are not present in this one line that was chosen for the genome project. So the question is, what are all these genes? And what is their contribution to quantitative trait variation? And that's some of the questions that we're now asking in our research. So now we can think about recombination rates, and this kind of like ties in with Jody's presentation. So this is rho. So this is like the historical or ancient recombination inferred from all of the SNPs. And as you see here, recombination rate increases from the centromere towards the telomere. Here's the centromere, and you can see it increasing towards the telomere. So in this region, there appears to be recombination suppression, but as you get closer to the chromosome arms and closer to the telomeres, recombination rate is actually increasing across the genome. And one key to point out is this is the ancient recombination rate that has happened in the history of the population. So by comparing the 90th versus the 10th percentiles, this varied about 12-fold for rho. So we also wanted to compare rho, which is, like, as I said, the ancient recombination, to the recent recombination rate that has happened in the Namm population. So what we found is an R square of 0.56. So coming out of Brandon Gaut's work, where he's done some he's basically been able to do testing of genomic SNPs to tell that these SNPs could be millions of years old. So what this means is that these recombination patterns tend to be stable over time. And if the age of these SNPs are actually very, very old, then these patterns are highly conserved through like the whole population history of maize. So the patterns of recombination that we're seeing now are kind of like the same patterns of recombination that have been happening like 10,000 years ago, or possibly even older than that. Excuse me. So when we look at Namm recombination, so this is the recent recombination, and we're checking out each chromosome, it becomes very dramatic then. So the 90th versus 10th percentile vary 28-fold for the recent recombination. And if you look at the centromeric regions, which are right here, there's not very much recombination going on at all. And almost the majority of the recombination is happening in the actual arms, close to the telomeres. So more than 95% of the recent recombination is restricted to slightly more than half of the genome. So there is like a whole other half of the genome that's only experiencing 5% of the recombination in the maize genome. So then the question that you should be asking yourself, how do you use this information and apply it to maize breeding programs now? So this is just an example of maize chromosome 1. If you look at the paracentromeric region, you can see a large region where it's completely recombinationally suppressed. And just to try to get an idea of how large these regions are, so all chromosomes, all 10 maize chromosomes, had a paracentromeric region of 60 to 113 million base pairs. So like when you think about 130 million base pairs, that's almost the size of an Arabidopsis genome. So this is tremendous, like, regions of the maize genome where there's no recombination happening at all. What we also found kind of shocking is these highly recombinationally suppressed regions contain 20% of the total genic fraction. Okay, so there's a lot of diversity in these pericentromeric regions of the genome, but they're just not recombining very well. So the two main correlated drivers of Namm recombination are, as you would expect, the location on the chromosome. Okay, as you get closer to the telomere, recombination is increasing. As you get further away from the telomere, recombination is decreasing. And the repeat content also has a big impact on recombination rate as well. Meaning the higher amount of repetitive DNA, the lower amount of recombination in that region of the maize genome. And then, so to even take this much further, we can compare it to pi. So we had kind of looked at pi in like my very first slide, which varied from 1 to 1.4% in maize. So the genome has many putative large selective sweeps based on a reduced pi. Okay, so these are basically regions of the maize genome where there's a low amount of genetic diversity. So the question is, why? So on average, pi was 0.66%, and the reason why that isn't closer to like 1 or 1.4%, if you recall, because we're trying to use all high-quality SNPs. When we're using our machine learning approaches to call SNPs, we're losing rare SNPs, and that's what's actually driving down pi. So just to try to give you an idea, what do I mean by a selective sweep? So this is, in a sense, a reduction of diversity from either artificial or natural selection. But as you see here, and it actually lines up really well, there's a strong relationship between diversity and recombination. And this recombination, just as a reminder, is rho. So this is like the ancient recombination. And you can see the peaks and valleys actually line up very well. So the correlation between rho and pi is possibly driven by selection and hitchhiking, and it had an R square of 0.33. But what we can clearly draw from this is that regions of low recombination retain low diversity. So we could also compare the correlation between the Namm recombination rate, which is the recent recombination rate, and pi. Oops, sorry, got ahead of myself. And that has an R square of 0.37. So we wanted to see, okay, what is the driver of these patterns in the maize genome for pi? So we then found out that, okay, pi was correlated with both the recent recombination R and rho, the ancient recombination, but then it was nearly independent of divergence from sorghum. So this then indicates that regions of reduced pi have been targets of selective sweeps. So of 148 intervals showing less diversity than the maize domestication gene TB1 at 0.18%, so not all the reduction were near the pericentromeric regions of the maize genome. In fact, of these 148 selective sweeps, 37 were in high recombination regions and 111 were in low recombination regions. But are these truly selective sweeps? So one caveat here is that these selective sweeps may not be associated with domestication, but selection in the ancestor of maize, teosinte. So these are just these long-term or very old patterns that we're now seeing in present-day maize. And these sweeps, as we heard from the previous talk, could be caused by drift as well. But one thing that's really important here is the investigation of the function and the adaptive importance of these regions will be very important in future maize research. And I think what Jody kind of touched on in his talk And it's very, I think, important point when you think of breeding and the maize genome now, is that breeding, i.e. selection, has had to work against very strong recombinational suppression in the maize genome. So this is my segue now. Okay, so that was kind of the maize hat map vignette. And now what we want to now do is actually use all of these SNPs, not just to like characterize the genome patterns of maize at the level of a population, but we now want to use these SNPs to find genes that are controlling complex traits. So one of the complex traits that I'm very interested in is carotenoid, which is vitamin A. And these are the 25 maize founder lines, and you can see that they vary for kernel color, which by that you can imply that they also vary for total carotenoid content and other of the composition of total carotenoids. So using the HapMap1 for vitamin A grain content, so maize accounts for 15 to 56% of the total daily calories in certain Africa and Latin American countries, and it is estimated that 250 to 500,000 children afflicted with vitamin A deficiency become blind each year, with about half of them passing away within 1 year of becoming blind. So this is a very serious issue worldwide, and our favorite carotenoid, beta-carotene, is converted to vitamin A in the human body. But one of the challenges here is a very small percentage of varieties have naturally high carotenoid levels, and some African lines have as low as 0.1 beta-carotene, but we're targeting 15 for our breeding efforts. And this is in parallel with Harvest Plus as well. So just kind of a proof of concept here. So we have now begun mapping actually both carotenoid and tocopherol traits. Tocopherols being vitamin E in the maize Namm population. So in this past year, there's been one grow out of the Namm population that we've run almost 8,000 HPLCs on. And I can tell you that is not high throughput. Okay, it's very laborious work, but it has to be done. Okay, so so what we're seeing here is some plots, and I'll kind of bring in the pathway now. So here, this example was actually shown in a previous talk. So here we have LIKE, which is lycopene epsilon cyclase. So what this gene is, it functions at a branch point here, and this work was published in Harhays et al. In 2008 Science. There's 4 different haplotypes of this gene here. So if it has a weak allele, what happens then? Most of the lycopene goes down this side of the pathway here, okay? So in a sense, it's like changing the flux from this side of the pathway over to the right. Why is that important? Because this side has beta-carotene, which has the highest amount of pro-vitamin A activity in the human body. So we want to increase, for the most part, the amount of beta-carotene in the maize grain. And what we've shown here in the maize Namm population, so first we mapped QTL with the approximately 1,200 common SNP markers, and then we used the GWAS approach, in a sense using all the ancient recombination with the HapMap SNPs. And as you see here, we clearly hit that gene. So this SNP is right in that gene. So this is great confirmation of all that hard work of running all those HPLCs. We actually find the gene that we should for this trait. One other thing that I would like to point out is this one hit up here on chromosome 7. This is a gene, but it's actually what we're referring to as an unknown gene, meaning that it's outside of the biosynthetic pathway. So by using this Namm approach, we're able to find new genes that are outside of the combinant pathway, because this approach here was taken with a candidate gene approach. So if it's a candidate gene approach, then you only know what you know from the pathway, okay? But with this approach here, we're now learning what is the true genetic architecture of traits such as like carotenoids and tocopherols. And now we're trying to tie that into breeding programs into with Harvest Plus into the third world. So now we're going to change gears. I'll put on my cotton hat now. So since I showed an example of maize, all its phenotypic variation, I can now show an example of cotton with some of its trichome variation. And it's actually been a really challenging change from maize to cotton, because maize I consider something that can be phenotyped very easily, where cotton not so much. It's basically a perennial tree species treated as an annual, and it has a lot of challenges, such as when you're counting nodes, you're like, what is the main branch I should be looking for? So there's a lot of challenges, but it's been fun nonetheless. So now I'm going to kind of concentrate on high throughput phenotyping. So why is this important? So there's a challenge to increase cotton yields in the face of climate change and diminishing water resources. I think most people in this room would agree that the genetic improvement via modern plant breeding is the most sustainable and economic approach to meet this challenge, and that the development of superior heat and drought tolerant cultivars has been slow, and I will actually replace that word with painful. So this breeding process could be improved quite rapidly with new ways to conduct phenotype to genotype. So after me, there's going to be some great scientists talking about GS. So that's one great way. But this approach is just going to be concentrating on the phenotyping. And as you heard earlier, precision phenotyping is very key when we're thinking about highly polygenic traits. So why, whoops. So high throughput phenotyping. So this is our sensors and our platform and vehicle. So right now we're able to do infrared. Okay, so let me just kind of position you here. So we have infrared thermometers, which allows us to do canopy temperature. So infrared thermometers are positioned right here. Ultrasonic transducers, which allows us to do plant height. And those are positioned right here. And this is a crop circle. So this is a crop canopy sensor and detects in 3 band wavelengths. And that allows us to do vegetation indices. And we also have a GPS RTK antenna up here. And this is a GPS RTK receiver and radio right here. So this is our high clearance tractor that we call it. It has an average speed of 2.82 kilometers per hour. Right now we're doing it at 1 hertz, but we're actually going to crank that up this year to 20 hertz, so we can get a massive amount of phenotypic data. And this is how we log everything here with these Campbell data loggers. So this is kind of, in a sense, like a test case. Will this actually work? So here we have is my cotton field. So I have like a helicopter that takes a photo of my field every 2 weeks, and this is one. The one was taken in August. So one great thing about Central Arizona: clear skies, hardly ever rains, but it's always hot. So it's a great place to do drought and heat testing in the U.S. And just as in population, so basically when I was hired there, I went to my seed room and I'm like, okay, what do I have in my seed room? Okay, I found a recombinant inbred line population that had a lot of seed, so that was going to be my actual test population that I was going to evaluate with high throughput phenotyping approaches. So my treatment here is 100% ET replacement, which is wet, and then half ET replacement, which is my dry treatment. And as you may expect in Arizona, everything has to be irrigated, and here we're growing it on drip irrigation. And what we have here is a heat map of the field which we generated from the tractor IRTs. Down here, this is the dry, rep 1, wet, rep 1, dry, rep 2, and wet, rep 2. So the greener it is, the hotter it is, and the bluer it is, the cooler it is. One thing I would want to point out is I ran an EM38 across the field to look at the electrical conductivity of the soil, and I found this region of the field had higher clay content. Okay, so that means when I'm calculating BLUPs, I need to use row and column and take that EM38 information into account. So when you're doing drought testing, you really need to not only know how you're going to manage your crops, but you need to know your soil profile as well, because that can create tremendous headaches down the road. So just kind of proof of concept here, we saw great time by treatment interaction for canopy temperature as we would think, and this is just BLUPs, and this is like averages at 7 a.m. here, 10 a.m. here, and 1 p.m. here, and this is the ambient air temperature. Yes, you see it gets very hot in Arizona. And as you see here, the dry is continually increasing, and this is at 1 p.m. So having a tractor is really great because we can have someone just drive through the field at like 7 in the morning, 10 in the morning, and at 1 p.m. So for this field test, it took about one hour for each like run when the driver is driving through the field. And that, as I said, this basically confirms tractor-mounted IRTs can detect temporal changes in canopy temperature. The treatment effect is strongest in the early afternoon. One thing I would like to try this year is driving around the clock, but I haven't been able to find a driver that would want to do that. So if there's anyone in the audience that would be up for driving 24/7, just maybe 2 or 3 days straight, call me. All right. So this is kind of like the captured phenotypic variation for canopy temperature in the recombinant inbred line population here. And this is one parent TM1 and other parent NM2416, and they hold the same rank compared to the wet and compared to the dry population here. And this is at 1 p.m. and this was in August. So it's actually heritable, which was a great relief for me. It isn't great, or I should say it isn't as high as I would hope it to be, but is at least something that could be used. So it had the wet was 0.52 and the dry was 0.58, and there was two reps for each treatment, just as a reminder. So how repeatable is this? So on the x-axis here we have day 224, so this is like 1 p.m. dry minus 1 p.m. wet versus day 217, 1 p.m. dry minus 1 p.m. wet. And it seems to be fairly repeatable. Obviously, I was hoping that would be higher. But what we can kind of say is that, yes, if you're doing tests for heat and drought in Arizona, it's almost the same every day. So we can get a very consistent phenotypic response to heat and drought stress. And about half of the reals had a change below 5.4 degrees Celsius. So now we're going to talk about NDVI, and just to give a background on what NDVI is, this near infrared minus red divided by NIR plus red, and these are the wavelengths here of the NIR and red that we're targeting. One really cool thing with this cotton population, as it gets more stressed, and you can kind of call this the droopy index, is the leaves in the afternoon begin to wilt. So what's happening as those leaves wilt, more soil is being exposed through the canopy, okay? So what then that allows us to calculate a wilting index, which is basically NDVI PM minus NDVI PM dry. So this is wet, dry, and this is wet right here. So as in a sense, as the greenness is dropping for like a certain plot, that means more soil contamination is coming up and it's being read by the crop circle. So in that case, the plants are very stressed and the leaves are very droopy and they wilt very heavily. And this is just one plant that's obviously very wilted. So as we would also hope, we saw a nice time by treatment interaction for NDVI, and this is at 10 a.m. and then you can see in a sense, the value of greenness dropping at 1 p.m. And it seems to hold pretty constant for the wet plots. But what I think is maybe happening here is that there's actually a trait in cotton that it can like the leaves like track the sun. So I'm thinking this may be sun tracking of the plants here, and that's what that changes, but I haven't confirmed that yet. So tractor-mounted canopy sensors can detect temporal changes in canopy geometry, and the heritability estimates for this trait were actually higher. So for the wet, it was 0.79, and for the dry, it was 0.59. And this also was highly repeatable. Day 224 at 1 p.m., this index, and day 217, this is 1 p.m. index, and it seemed to be fairly repeatable. And kind of the same conclusion here. There's like a consistent phenotypic response to the drought and the heat stress over the growing season. And this is just half the rills have a index below 0.13. So, and then I wanted to kind of see, well, if the plants are beginning to wilt more, they'll be hotter. So I wanted to look at the relationship of the change in canopy temperature and the index as the plants wilt. But one thing to point out here, though, is as the plants wilt more soil is going to be exposed. So that means as the IRTs are driving over top of the plots, there's going to be some more soil contamination coming through, and it's hitting the IRTs. So that's going to artificially inflate in a sense, the canopy temperature. So that's something that we're trying to work through right now. If there's anyone in the audience that have some ideas on how to work through that, that would be great to talk later. But that's kind of where we are with that. So increase in canopy temperature results, and plants begin to wilt more. So kind of the conclusions from this, we were able to develop a field-based high-throughput phenotyping approach to rapidly phenotype hundreds to thousands of plots for canopy temperature, and what I kind of refer to as my droopy index, but obviously it's just the rate as when plants wilt. The canopy temperature and the wilting index are moderately to highly heritable as well as repeatable on a temporal scale. And what we kind of see this, or foresee this as a tool to map QTL. And what we would like to do in the future is use it to select stress tolerant lines, but we're not quite there yet. And then as time goes on, I'm now kind of tying in yield and fiber quality information as well for all of these tested plots. So where are we going for the next 5-year cycle with my program? So we want to continue this HTP platform for it to be more precise. And so obviously heat and drought are important areas in where we grow cotton in Arizona. I also want to target yield. I have received funding from Cotton Incorporated to create a cotton Namm population. Yes, I haven't learned my lesson. So, but this one's going to be a little smaller. So we're thinking maybe 3,000 lines and possibly more parents and like smaller population size. But we'll see how that plays out. And GS. So since these traits are highly polygenic, I think being able to combine GBS in cotton, which we've actually have been able to perform quite well. This is in a project with Jesse Poland at Kansas State, and we've been able to do GBS in cotton. So if it works in cotton, it'll work in everything else, trust me. So as we move forward, we now are beginning to have like this GBS platform, if you will. We're creating more populations to like capture variation and see its importance under heat and drought. And we also want to tie in GS as time goes on. And this is from my Maize life, all the folks at Cornell, Cold Spring Harbor, Purdue, UC Davis, and Michigan State. And this is from my cotton life, all the people from my lab, University of Arizona, New Mexico State, USDA-ARS, where I work, and other people from satellite sites at the USDA-ARS across the US. I thank you for your time. Ah. One thing I would like to point out with this is we tested the contribution of like pi, like R, the Namm recombination rate, the gene content, all in that area, and we found that recombination rate was the major driver. But one key to kind of point out though is a lot of these factors are highly correlated. So it's very challenging to tease them all apart. Now, on the issue of not having enough coverage in those regions, there are actually I don't know the exact quantity of genotypes coming from that region, but that region has been covered and pretty well characterized. And with the HapMap project, I should say, that region has been pretty well covered. And there's now HapMap version 2, which was whole genome shotgun of these 27 lines that have also covered that region, and that trend holds up as well. One point I would like to also bring up is when thinking about genotyping and breeding, and you have these regions where there's heavy recombination suppression, the thing that breeding companies can do very well is genotyping very cheaply. So they can create like massive populations and then just do seed shipping to find the rare recombinants in that region. Did that answer your question? Okay. Yes, Jimmy. Yeah. Uh, that is kind of a loaded question, Jimmy, but I will answer it to my best. Obviously, there is a lot of subjectivity and preference that goes into choosing lines by a breeder who's been doing it for 20 or more years. What I see this as is a complement to what a breeder is doing out in the field. I don't see this as a complete replacement. No. Mm-mm. Anything else? Yes, Aaron. Okay, so what I can say is canopy temperature is very important to cotton. Okay, but cotton has like a long growing season. Okay, so to really get a strong, well, I should say to like actually have it like tightly linked to like yield or like fiber quality, you'll have to harvest like certain portions of the plant, right, over the growing season, and then see what like the canopy temp was at that time, because as the plant is putting on more and more flowers, so that's kind of the challenge there. I mean, if you just take like canopy temperature from like one fixed point when you're driving through the field in like August, it won't always be highly correlated with what you harvest at the end of the year, because obviously with the change in temperature and precip over time, then there's certain, I'll say like portions of the plant that you should like harvest, and that will be more like tightly linked to like canopy temp at a certain time point. So to actually kind of like tie that with like end in yield, that's kind of the challenge where we're at right now for a plant like cotton. Possibly something like maize, it may be easier, but I could be totally wrong. Yeah. Wait, you're saying comparing to handheld? Oh, okay. So that's a great question. So, okay, yeah, that's fine. So this question is, how does like the high throughput tractor platform compare to like handheld, right? That's the question. Okay, so one thing with like using say a handheld for like chlorophyll content, say you have like 100 plants in a row, which is like what we have for cotton. You have to think about the tremendous amount of time it would take for one human being to actually phenotype, or to like test every plant within that row. With the tractor, we're able to do like all the plants in a row. But to go further is we need to have like a head-to-head comparison, such as like chlorophyll content from the tractor and chlorophyll content from the like a handheld. But so that's kind of one thing I would say is like a real advantage of the tractor approach. You're able to like scan all the plants in a row. And one thing to point out is so when I'm processing all of this in like ArcGIS, is I'm cutting like the 5 foot off of each end of the plots, and then I'm checking out for outliers as well. But it's a lot easier to check for outliers when you've like scanned like every single plant than when you've only done like 4 plants or 2 plants in a plot. Okay. So there's this cool instrument called a LiDAR, and we're going to begin using that this year to do canopy geometry in cotton. So when you actually look down a cotton row, there's a lot of various branching patterns that you can see by eye, but actually to go out there with like a, I don't know, with a tape measure, it takes a lot of time to see what all those branch links are, right? So what we're now doing is trying to do canopy geometry. So then we'll have like the plant height and like the branch angles and like the width of the plants as well. And it's kind of cool. I guess a lack of word for it, and I think it's a word to kind of use. You kind of create like a cloud of it, and you have like all these points. So you in a sense can like recreate the shape of the plant. Yeah.
Okay, so lunch, we should have no shortage of things to talk about.